Okay, so we have gotten as far as the role of calcium inside of a muscle fiber cell, which is to bind to troponin, lift tropomyosin off of actin so that myosin can bind to it, and myosin can pull actin toward the M line to shorten the muscle, aka a contraction. The last bit of this story is, how does this myosin molecule, myosin head, get the energy to pull? We've gotten as far as how it can attach to actin, but how does it actually do the pulling? That's the end of this story. So I'm just going to draw a vertical line here and describe in that space how this molecule obtains its energy. Way back at the beginning of the semester, we talked briefly about an ATP molecule, which I'm reminding you what that is. I've just done a quick drawing of an ATP molecule. The significance of this is, how does this molecule transfer energy? What's the big deal about ATP? Well, just to remind you, if we cleave or break this terminal phosphate off of ATP, which stands for adenosine triphosphate, 1, 2, 3 phosphate, if we break that terminal phosphate off of this molecule, this becomes ADP, adenosine diphosphate, plus P, phosphate. The physical breaking of this bond transfers energy from this molecule to another molecule. It's chemical energy becoming kinetic energy. So that's what's going to happen here. So we're going to draw another myosin molecule. There it is. This is the myosin. Here's the globular head of the myosin molecule. Now I'm going to quickly draw actin. I'm going to leave all the tropomyosin and troponin stuff off of it right now. What we're interested in is how this molecule, after it attaches to actin, can actually pull it, gets its energy. Well, the secret is in this molecule, ATP. The whole reason that we eat food and breathe oxygen, mostly, is to make this molecule. It's the fuel that does so many things in our bodies, and this is a great example of that fuel being used to do something. So I'm going to just use green to designate ATP. Here's an ATP molecule that's in the cell, in the muscle cell. These ATP molecules bind to the myosin globular head. So the ATP attaches to this myosin head. So I'm going to draw that. There it is. We have an ATP molecule attached to the head. Now recall, when we break this terminal phosphate off of the ATP molecule, it has the ability to transfer energy from the ATP molecule to something else. That's what's going to happen here. If we cleave this terminal phosphate on this ATP molecule, what we end up with, as we stated earlier, is ADP plus P. There it is. We just broke that terminal phosphate off of the ATP molecule. Both of them are still attached to this myosin head. But when we break here, it transfers energy to here. What happens physically when we do that is this myosin head goes from this position to this position. So I'm going to draw that with a little arrow pointing down. Just to reiterate, ATP attaches, we break the terminal phosphate off of ATP so that it becomes ADP and P. When we do that, the myosin head does this. It's like pulling that thing on the end of a gun, like the cocking a gun so that it can fire. That's what we've done. I have to draw ADP plus P because they're still attached. The breaking part of the terminal phosphate is what gave this energy to move. Now, after this occurs, myosin attaches to actin in that open position, reaches up, grabs actin, and it pulls it this way, toward the end line. So we've exposed the active sites on actin with our calcium story. Now we're going to give this myosin head energy. ATP molecule attaches, we break the terminal phosphate, that gives it energy. It reaches up, grabs the actin molecule, and pulls it toward the end line. Now here's the very end of this story. Even after myosin has reached up, attached to actin, pulled it toward the end line, what happens next is the ADP and P leave the cell. They drop off. They dissociate from the myosin head. But, and this is critical, this is very important, the myosin head remains attached to the actin until a new ATP molecule attaches to the myosin head. So I'm going to draw that last bit of this story. We've gone through, given myosin energy by cleaving the ATP. I'm going to draw a myosin molecule up here. It has pulled the actin molecule toward the end line. ADP plus P are still attached when it pulls. After it pulls, the ADP and P drop off, dissociate from the myosin head, but this remains bound to actin until a new ATP molecule attaches to the myosin head. Only after that point does myosin release, and then the whole process starts again. Myosin releases after a new ATP attaches to it, and then we break off the terminal phosphate, giving it energy. It reaches up, grabs, pulls it. ADP and P drop off, stays released until a new ATP molecule attaches to it, and then it releases, the whole mechanism releases, and it just keeps repeating itself as long as we want a contraction to occur. Myosin not being able to release from actin is what causes the process of rigor mortis when somebody is deceased or dead or passes away. When somebody dies, their muscles contract into a state of continuous contraction called rigor mortis. And those contractions don't release because the deceased person's cells aren't making new ATP molecules